maintenance type things to touch on. I discovered in reviewing the statistics, which the exam service sent me late this afternoon, that there's at least one question that was miscoded on the answer sheet given to the exam service. And therefore, we're going to have to rescore the exams. So I can't give you final statistics and so forth. For the most part, it looks like things are fairly good, but I can't give you a very precise one until we have corrected that one and double-checked the rest. There's one question on which a case was made for 2 answers, and that was factored into the scoring, so people don't need to separately request that. There was one on the, I don't have the question number, but it was about things in carcinogenesis, and instead of increasing telomeres, it said something about eroding telomeres, and phrased the way it was, there were in fact 2 correct answers. It only affected about 8 students. So, more looking to the future. All right. Looking to the future, it's hard to believe it, but we are approaching the final exam. A week from tomorrow will be the final exam. The handout you have has the wrong auditorium number. I'm not quite sure where in the line of communications the 3 got into it, but it should be auditorium 2, SPI. Oops, what is this happening? Back up. Because it's highly desirable to have enough seats for alternate seats for a final exam, we are split between 2 places. The number of seats we have available will be enough plus something like 8. So it is very important for you to go to the correct auditorium. In this room, if your last name begins between A and M, come here. If your last name begins between N and Z, go next door. It's not very far away. But we will be asking the RAs, the proctors, to look at some kind of ID to confirm you're in the right place, because we won't have room to accommodate very many strays. I don't like that. I'm sure you don't like that. But I have not yet had my requests for a single large enough final exam room agreed to by the campus, by the administration. Yes?
Is it odd 2 or odd 3 on our notes?
It says odd 3 on the notes. I think you just slipped in, perhaps. Yeah, I just said that somewhere in the line of communication a mistake was made, and it's actually odd 2. And I'm going to send out an email in addition to make sure that people who aren't here still don't get lost. But do have some ID with you. I'm not fussy about it necessarily being university ID, but it is helpful to have photo ID if at all possible. But it has to be some kind of ID. If you don't have ID at the final exam, we will ask you to wait until everybody's seated, and we will only seat you if there's space. So it really is worth it to remember to bring some ID. I've mentioned before that in addition to the review sessions this week, you would find it valuable to look back at the past exams from this year and to look at those questions as review questions for the final exam. All right. So what the point of this last week of overview is a chance to revisit the things we've talked about. And in many cases, students find that they suddenly start to gel, that there are things you've heard more than once perhaps during the semester, but they suddenly start to connect and to make sense. And so what I hope this week will do, beyond anything it does for the final exam, is give you a chance to have that kind of synthetic coming together experience of biochemistry, and also to ask questions on things that you still don't quite get. And so if in the process of going through these things, you hit across that thing that always kind of graveled you, please raise your hand. If you didn't quite get it yet, someone else didn't either. So do feel free to ask questions as we go along. And I apologize for my voice, but this is all that's come back. So we're going to be starting by talking about biomolecules and their variety. The extraordinary variety of biomolecules comes from a very small number of atoms. And so the greatest mass of the biological molecules comes from just these 6. Excuse me. Carbon, obviously, because they're organic molecules. Nitrogen and oxygen. Sulfur, hydrogen and phosphorus. Hydrogen in large numbers, although not a lot of mass. These specific elements appear over and over again because they're among the most abundant. Things like cadmium and molybdenum, there's just not very much of it to build life out of. But also because carbon especially has properties that are unique to carbon that allow it to form large, complicated molecules with a lot of variation in structure. The closest companion to carbon, which would be silicon, we know cannot form a comparable level of complexity. These four elements can be related to each other along their electronegativity, the least over here and the most electronegative there, hip casino. And the reason this makes a difference is that the ones that are more electronegative are going to have the lion's share of the electrons. So in any bond between hydrogen and oxygen will have the larger share of the electrons and will have a partial negative charge. It also means that there's a finite number of functional groups that need to be learned in order to have sort of the chemical vocabulary to think about biochemistry. There are four basic kinds of biological molecules with slightly different purposes and strategies, if you like. And those four groups are the amino acids, the carbohydrates, the nucleotides, and the lipids. Here's a sample for an amino acid. This extremely simple core with an amine group and an acid group, and then a side chain of some kind or another. The carbohydrates get their name because it's roughly carbon plus water, which reflects that carbohydrates are polyalcohols. If we look at glucose here, we can see all the alcohol groups hanging off, a little more obvious in the linear representation. Carbohydrates can actually be a ring or a linear form, and how much of the time they spend in the ring or the linear form depends on the particular sugar and to some extent on the environment. Nucleotides are more complicated than either of these two. They combine some elements of one of them because for a nucleotide to exist, you'll have a ring sugar, a ribose or a deoxyribose. You'll have a base of some kind. There are only, fortunately, 5 of those, 4 in RNA, 4 in DNA, so ACTG or ACUG. And then a tail of some kind of 1, 2, or 3 phosphates if it's a nucleotide. No phosphate at all, a nucleoside, a sugar. And then the fourth major group are the lipids. These are the kind of hydrocarbons of various sorts, sort of, kind of. They're structurally fairly diverse in terms of exact position of the double bonds, exact bond lengths, the exact head groups and stuff. They share the fact that they're poorly soluble in water. That's probably the defining characteristic for lipids. And many of them have what looks like a hydrocarbon structure as a significant part of the chain. You have here cholesterol, which is a somewhat unusual lipid, but it's a very important one with its fused rings. But it's still going to be more like a hydrocarbon than not, since this little alcohol down there is its only polar component. So from those 4 kinds of small biological molecules, we can build up bigger molecules. We can build up polymers from the monomers. I will mention that you can apparently make polymers out of lipids. This is not a common behavior, and I'm not going to say anything more about that. The polymers we're talking about are from the other 3. Proteins are certainly one of the most conspicuous of the polymers. They're polypeptides, so they're going to be polymers of amino acids head to head. The polysaccharides, multiple sugars in a group, and then the nucleic acids, DNA, RNA. And what these polymers share is that they're going to be built from subunits. You can see sort of a schematic for polymerization. Many of them are linear. The polysaccharides, not so much so. And information of some kind or another, the polypeptides and nucleic acids are more information dense, but polysaccharides do carry some information, especially on the surface of cells. For polypeptides or proteins, we know that there are 20 amino acids that are used during synthesis. A few others appear later on after the fact. Each of those amino acids sorry about that. Each of the amino acids then has a unique shape and the variability in its structure. So what these side chains allow for is extraordinary functional diversity in the polypeptides. By contrast, polynucleotides have only the 4 bases, only 2 major forms. They're almost always linear. They carry information in the richest sense. That is, they're intended as linear arrays of information to carry things. Although, as we know, there are also times in which nucleic acids can act catalytically or structurally. And there are not nearly the diversity of structural forms that we see with the polypeptides because the much simpler complement of bases to work with. Polysaccharides, I'm not quite sure if I'd still say usually homogeneous. The bulk ones tend to be. So cellulose is pretty homogeneous. It's all glucose polymer. Starch is homogeneous. It's all glucose polymer. Extracellular polysaccharides are not particularly homogeneous. They're great for fuel storage. I just mentioned starch as an example of that, or glycogen in us. They're great for structural support, cellulose, for example, in plants. The starches and cellulose are principally linear, some branching in starch and glycogen. Extracellular polysaccharides more extensively branched. It's principally these heterogeneous extracellular polysaccharides that are going to carry information. But overall, polysaccharides don't really come close to polypeptides in the structural variety that they can accomplish. Any questions before I go? Yes?
At the top of the slide, when you said they were often linear, you said not so much polysaccharides, and then you just sort of did frequently linear. So could you specify that?
I guess, so I have frequently linear there and often linear there. I guess it sort of depends on whether you're thinking about categories or mass. By mass, cellulose is going to be the greatest mass of carbohydrate because it's the walls of plants, and it's linear. If you start to count numbers of things, there's a greater number of extracellular polysaccharides than there is of starches or cellulose. So I guess what that would say is that frequency adjectives here are probably not very helpful. Proteins are synthesized as linear molecules. Nucleic acids are synthesized as linear molecules. Of the polysaccharides, if they're extracellular polysaccharides, they may well be synthesized from the start as a branched molecule. Among the proteins, you occasionally get one branch. So, for instance, if you look at insulin, you could think of it as a branched molecule because it's cross-linked into the middle of a side chain. In the case of nucleic acids, you've got a few that have some kind of a branching structure to it. But in the case of the polysaccharides, we know, for instance, that glycogen is branched. We know that starch is branched to different extents. Surface polysaccharides are substantially branched. So does that help unpack that a bit? Okay. Any other questions on that before we move on to this? Okay. And thank you for asking. Can people still hear me? Yeah? Okay. In addition to the variety of chemicals that goes into making life, there is a little bit of thermodynamics that's absolutely essential to have a grasp on it. We can capture some of the important properties of a reaction in a property called delta G for free energy, or Gibbs free energy. And for any given reaction, that free energy change may be positive or negative. And in a reaction like the first one shown here, of A going to B, where the Gibbs free energy change is a negative delta G, we call that reaction a spontaneous one, meaning it will go forward without the input of energy. If it has a positive delta G, it will typically not go forward spontaneously, but it can if it's coupled to one that would. And so for the example we're showing here, if we look overall from A to C, overall the reactions from A to C still have a negative free energy change. So overall, we would say from A to C is spontaneous. And that free energy difference is something that shows up as a pattern in metabolism. So for instance, if we're looking at the side of making things, fixing carbon dioxide to make something interesting, like even a sugar, to go from a fully oxidized carbon to a partially reduced one is not spontaneously favorable. And we know that in the wild, in the environment, energy is put in the form of light. But this partially reduced carbon is a relatively energy-rich compound compared to at least this. If it's oxidized, it can release free energy, and therefore its oxidation can also be used to drive other unfavorable reactions. And this is just reminding you about the oxidation states of carbon. The rule of thumb here is looking at the number of bonds the carbon makes to either oxygen or to hydrogen. A carbon-oxygen bond, the carbon is deemed to be have an oxidized state from that one. A carbon-hydrogen bond, the carbon is deemed to be reduced. So in carbon dioxide, all 4 bonds are to oxygen. It's fully oxidized. You're not going to get a lot of free energy out of it. In methane, with all 4 bonds to hydrogens, it's the most reduced, the richest in energy, and that's why you can burn it in your stoves and cook your dinner over it. So the more reduced it is in general, the greater the free energy that can be released from it. Oxidation releases energy, and the products are usually lower in free energy. Well, all of this beautiful chemistry doesn't happen out in the wild. It happens in cells. And one of the key things that has to happen is carbon has to be fixed into an organic form. That's one of the triumphs of living cells. And the great bulk of that is accomplished through photosynthesis. This was first put on a major industrial scale through the blue-green algae or the cyanobacteria. And that's partly because they were able to finally find a great source of electrons, and in the process they released oxygen. So the oxygen in the atmosphere is there because plants made it for us. And the blue-green algae made the Earth safe for eukaryotes. So their photosynthetic capacity is very important. All of this started at least 3.5 billion years ago from the earliest traces we can find. The simpler cells, not all that simple, but simpler cells we call prokaryotes. They don't have nuclei. They're before the nucleus. Come in 2 major flavors, the eubacteria and the archaea. Whereas after there was oxygen in the atmosphere, after there was already a reservoir of prey to eat and things like that, the eukaryotes developed, and these are conspicuous for a number of things. They have organelles inside the cell with internal membranes. Now, some prokaryotes have internal organelles with membranes. In eukaryotes, the double membrane-bound organelles are uniquely eukaryotic. The nucleus, the chloroplast, the mitochondria are only in the eukaryotes. Some eukaryotes have a wall, some don't. The same could be said for prokaryotes. Eukaryotes come in a very wide range of sizes, from a micron to the length of a whale neuron, so it's a wider range of sizes than prokaryotes. They can accomplish more complicated morphology. compare a sequoia to an E. coli. But because of this, eukaryotes are less able to live under the really far reaches of life. You don't find eukaryotes living in sulfuric acid. You don't find eukaryotes living in boiling water and so on. And the nucleus is the clear hallmark of a eukaryotic cell. It is the organelle that holds the information for the ongoing life of the cell. There are a few eukaryotes who don't have mitochondria. There are no eukaryotes missing nuclei altogether. Excuse me. Any questions? Well, when we look at cells, we discover that an awful lot of cells isn't even organic molecules. Anywhere from 70 to 90% of the cell is water. And water is a hallmark of life as we know it for a number of reasons. Probably any life, but that's a separate question we can talk about later. It's essential. Excuse me. As a solvent, it has a remarkably broad spectrum. As a solvent, the things that it can't dissolve contribute to the structure of proteins and membranes. That is, the things that don't dissolve in water that are hydrophobic then contribute to structuring other things. Water is also behind acid-base relationships, and the ability to gain and release protons is an important part of many enzymatic activities. Water is so essential to life that life is tied to liquid water. We're not going to find actively metabolizing cells without water. So that sets temperature limits. It also defines what kinds of chemistry are possible. And here's a structure of our essential little molecule. So you have your oxygen in the middle and your hydrogens out here connected by electron clouds. And because there are these two unshared pairs of electrons here, if you look at a ball and stick diagram of water, overall you're going to see that there's a partial positive charge on the hydrogens and a partial negative charge sort of sticking out the rear end of the molecule. So water is polar in its bonds. It's polar as a molecule. So it has a positive end and a negative end. And it has the capacity to form hydrogen bonds. Each of these partially positive hydrogens can form a tenuous but important hydrogen bond to a partially negative partner. In liquid water, that partially negative water partner is oxygen itself. And because there are these 4 points of contact, one water can go up to 4 bonds. And in a hydrogen bond, the electron is partially shared then between the hydrogen and something else. If it's well aligned, like this, it will be stronger. And this polar structure of water with its extensive hydrogen bonds contributes to these unique characteristics or highly unusual characteristics of water. That it has a high melting point, a high heat capacity. It's a wonderful solvent. When it's fully bonded, it makes ice, and ice is less dense than water and floats, so our oceans don't fill up with ice. This little graph gives you a comparison of the relative strength of several kinds of bonds. Here's a true covalent bond, which would be the shortest of them and is the strongest of them, meaning it takes the most energy to disrupt it. Here's an ionic interaction, which is not as strong, but is stronger than the hydrogen bond. The hydrogen bonds can form with oxygen, as we mentioned, nitrogen and sulfur. And although they are weaker than the covalent bonds, because they're so numerous, they end up playing a very important role. Weakest of all are the van der Waals. It's solvent character that makes it such a lovely solvent. I mean, it's polar character. So the negative ends of the oxygen can shield the sodium, and the positive hydrogens can shield the chlorines. And in a molecule like glucose, each of those alcohols offers two different partners for hydrogen bonds. And in a molecule that doesn't have a polar character, is neither ionic nor polar, with partial positive, partial negative charges, interactions with water can still be important through the hydrophobic effect. It literally means water fearing. There is no antipathy between a hydrophobic molecule and water. It's just that because it isn't polar, water is not free to orient relative to it the way it would with other molecules. And so the water that's at the most intimate, oops, I seem to have gotten out of the, there we go. Sorry, when did that happen? The water that's at the immediate contact with the hydrophobic group does not have the same freedom to orient that water elsewhere does. So if there are multiple molecules that are hydrophobic, if they end up in contact with each other, excuse me, less water is constrained, and the entropy overall is better. The water is more disordered. The hydrophobic things are more ordered, but overall, it'll tend to cluster. And this is the principal energetic basis for both the structure of membranes and for proteins, to a considerably lesser extent for nucleic acids. Now, in molecules that have any considerable extent to them at all, you could end up being amphiphilic. You could end up having one part of the molecule of one kind and the other. And here we see an amphiphilic lipid, where you've got a hydrophobic tail and a charged head. And molecules like that can sort out into lipid bilayers and into micelles. Here the advantage is that since the interior of that lipid bilayer is hydrophobic, compounds inside such a vesicle that are polar or are charged are not free to diffuse out. In addition to interacting with itself through hydrogen bonds, water can, in fact, dissociate into a proton plus and an OH minus, or OH3 minus, H3 plus, I don't know, OH minus, but doesn't do it very much. If we're talking about water at neutrality, 10 to the minus 7th molar H plus is not very much. So we use a negative log scale pH to talk about it. 10 to the minus 7th becomes 7 on a pH scale. So anything smaller than this is a higher concentration of protons and is more acidic. Anything larger than 7 is more basic. And this is a little reminder of the structure of some functional groups that functional groups are capable of. I'm going to take this off for a second. Excuse me. I don't think anyone wants to hear my nose blowing. I don't think I can suck on it and talk at the same time. Thank you for thinking of it. I'm getting some done. I'm doing the best I can. Thanks for your patience. So a carboxylic acid, here's the carboxylate. Here's an amino acid, here's an amino group, an amino ion. And as we go to the lower pHs, there are more protons. Those groups are protonated. We go to higher pHs, fewer protons around. Those have lost their protons. They're deprotonated. And in the process, the carboxylic acid has become ionized, or carboxylate, in going into the basic thing, has become deprotonated or unionized. We'll start thinking about some of the biomolecules, not just the sort of the background machinery. DNA and RNA have an awful lot in common. We can think about them together. They're going to have a ribose or a deoxyribose sugar. There's going to be a base attached at the 1' position of that, and there may or may not be phosphates off of the 5' of the sugar. There are primes in the sugar because the first numbers are taken up with the bases. And this is showing you some of those in a little more detail. Mononucleoside, monophosphate here, diphosphate, triphosphate. The adenine base, the guanine base are both fused rings. These are purines. The thymine and cytosine are pyrimidines, or single bases. And then the phosphodiester bond, because it's an ester to this direction and an ester to that direction, making the nucleic acids linear molecules. Here's a free 3' end with a hydroxyl. Here's a free 5' end with a phosphate. For that not to have a phosphate, there's something we need to remove, that phosphate group. A great majority of nucleic acids are found in circumstances where their capacity to make base pairs happens, certainly for DNA, to some extent for RNA. And we can see that the base pairing ends up producing this regular double helix. So it's a vertical helix, or a spiral that doesn't get bigger, if you want to call it that, in which one purine and one pyrimidine can make hydrogen bonds to each other. The phosphate groups are towards the outside, the bases are towards the inside, and 10 base pairs will give you one full turn of a helix. It's always a purine to a pyrimidine, A to D, G to C. And even in these base pairs, you should note that there are still potential hydrogen bonds left unoccupied, so that although they are well bonded to each other, there's still contact in both the major groove and the minor groove for other kinds of hydrogen bonding. That's how DNA sequence recognizing proteins can do it. bDNA is the most common form of DNA. It's the bulk of the genetic material most of the time. aDNA is an analog for RNA DNA double helices, which turn up in a number of places. It's not an exact model, but it's closer to RNA DNA hybrids than bDNA is, fatter, wider. And then there's zDNA, which is actually a left-handed helix with the backbone going up funky, and it is found frequently in regions that are called methyl GC islands, which may play a role in gene regulation. So although zDNA is not very common, it is of growing importance as we come to learn more about it. RNA is also capable of folding regular structures. We most often find RNA molecules that we make for ourselves as single strands. Viruses make double strands, but we mostly make single strands. But those single strands often have the capacity for base pairing within that single molecule. And here's an example of a transfer RNA where you can see the base pairing has contributed to that. There are other things going on in that thing as well. And so the complicated structure of RNA reflects the fact that RNA has more functions than DNA does. DNA is storing the information. RNA has a number of different roles that it plays in getting that information expressed. Any questions? So I mentioned the hydrogen bonding that can make the base pairs. Whichever base pairs they are, they're the same width, so it gives a very smooth packing to the double helix overall. The bases have considerable pi bond character, and so they can interact base to base through what are called stacking interactions, and it's actually the stacking interactions that give most of the stability to the double helix. Stacking interactions are sufficiently strong that apparently even a single strand of nucleic acid will tend to have a good fraction of its bases stacked one on top of each other because they are more comfortable that way. GC pairs tend to stack with a stronger affinity for each other than AT pairs, and so the more GC in a DNA, the more stable it is against heating. But it's not because there's 3 hydrogen bonds. That's a coincidence that's misleading. The relationships among the nucleic acids and protein has been called the central dogma. This is something that developed in the 60s, and it certainly organizes a lot of thinking in molecular biology. Nucleic acids have the capacity for complementarity. And the complementarity of DNA lets it replicate to make more DNA. The complementarity in transcription lets it generate a mirror image, if you like, RNA molecule. That linear sequence in the RNA can't by itself make protein synthesis. It has to have adapters of some kind in order to make linear protein. For the great part, it's one-way information flow. It comes from the gene, it goes to the RNA, it ends up in the protein. There are some exceptions. RNA viruses obviously make life happen without DNA, in some cases at all, certainly not as their mainstay. Reverse transcriptase. Telomerase can take a little piece of RNA and make DNA at the end of the telomeres. So this is a generalization, but not an absolute generalization. Any questions so far? Okay. And this is just a picture of that kind of complementarity. In the transcription, I pointed out the handedness of strands, that nucleic acids have ends that are different. They have 5' ends and 3' ends. And this is just showing you how the growing messenger RNA molecule is growing from the 5' towards its 3' end. Its template is being read from the opposite direction. The template is complementary to the message. The strand that lay across from the template strand is sometimes called the coding strand, because except for the T for U business, it has the same sequence as the messenger RNA. In order to generate that last level of complementarity, there has to be a way to take RNA to protein. We have 20 amino acids. We have 4 bases. One base could code for 1 amino acid. If you could take 2 bases at a time, you could code for 16 amino acids. So we know that it's a triplet code. There have to be 3 bases at a time read to code for an amino acid. What we didn't know, what didn't have to be from logical reasons, is that the triplet code was so beautiful. So in this drawing of the genetic code, it's bringing out the fact that codons of triplets are close to other amino acids, other codons that are for similar amino acids, so that the codons for leucine are close to phenylalanine. The codons for lysine are close to the codons for arginine, and so on. So this is really a remarkably robust structure to the code, and it allows accidents in replication or mutations to have less impact than they would if there's just a random code. This is a way of showing you how tightly connected different sequences are to each other. We've covered this fairly recently, so maybe I won't go into quite the same detail. I want to remind you that in us, not necessarily in all organisms, but in us, a good 3/4 of the DNA is never transcribed. It's in there, but it's not transcribed. Of the quarter that is transcribed, a lot of it never leaves the nucleus in any form. Of the transcripts that will leave the nucleus at some point, there has to be a lot of processing before it gets out. So there's a lot of what's going on in this transcription that we're not really understand yet. The stuff that gets out and translated, we know why the cell's doing that. They want the proteins. But there's some transcription going on in here that we don't fully understand why the cell bothers. But it means that you can have a stretch of DNA of, 250,000 bases transcribed into that big an RNA, end up with a processed message of only 6,000 bases. and only get a protein of 1,400 or so amino acids out of it. This is a diagram that's trying to reinforce the idea that the information flow is fairly simple from DNA to DNA or to RNA to protein, but that these processes themselves need the products of the processes. So it's a recursive relationship. This is looking at some of the things we spend our genetic information on. E. coli is one of the best understood organisms, and yet nearly 40% of its genes, we don't know why they're there. They're there. We don't know why. We have maybe 20% going for metabolism, daily bread sort of stuff, 5% for structure, you know, you build your house, about 8.5% for the core macromolecular metabolism. Transport's a big deal, getting things in and out, 10%. Regulation. So a lot of activities. Our current estimates are that the fewest genes a cell could get away with would be somewhere in the order of 2.5 to 300 genes. If we want to think about protein structure rather than nucleic acid structure, we have to think a little bit about amino acid structure because they're more complex than the nucleotides. Nucleic acid structure is so simple because of the base pairing. Protein structure needs to be complicated. It needs to be Byzantine, and so we need to think at least a little bit about the amino acids and how they're weird. We have some amino acids that are hydrophobic. That is, their side chains tend to avoid water, and they come from very small side chains like alanine to very big ones like tryptophan or phenylalanine. Some of them are just plain hydrocarbon side chains, like these guys. Some of them are aromatic, like phenylalanine or tryptophan. Methionine is fairly hydrophobic, although it does have its sulfur sort of tucked in the middle there. Proline has a hydrophobic side chain, but more importantly, it closes back on itself, which makes it sort of a kinky amino acid, and therefore it puts kinks into protein backbones. The other amino acids have some capacity to interact with water. Tyrosine is in here because it has the alcohol group, which certainly can interact with water. Its aromatic ring prefers not to, and it sometimes tucks itself into proteins so that the toe of the alcohol is sticking out. These are all the ones that are being shown as simply polar, neither charged nor hydrophobic. And then there are 4 that actually can be charged, not merely liking water, but having a full negative or partial charge. 2 with acidic side chains, aspartate and glutamate, both of which we'll see in multiple places in metabolism, and 2 with positive side chains, which are also important for protein structure. And this is just pointing out that a few of these side chains can undergo protonation, so histidine and cysteine are often found in active sites of enzymes because they can undergo protonation or deprotonation in regions close to neutrality. pKa's 6, 8.3 are close enough to neutrality that they can be part of enzymatic reality. Cysteine is also peculiar in its ability to make a disulfide bond that can cross-link proteins. They're no use unless we can put them together. Here is an amino group of an amino acid. Here's the acid group of an amino acid. If you, in your mind, take out a water molecule, you now have a peptide bond with a carbonyl oxygen and an amide nitrogen. The charge here tends to be distributed across these things in such a way that this bond, the peptide bond, tends to be a planar bond. That is, the oxygen tends to point directly away from the hydrogen or the amide hydrogen. And so the alpha carbon, the carbon at the center of the original amino acid, becomes a pivot point for all these things to twist around. That it can twist on that bond, it can twist on that bond, but the peptide bonds behave in a fairly planar way. And that means that there's going to be constraints on the overall structures that proteins can take. The primary sequence is simply the linear sequence, or the primary structure is the linear sequence of amino acids. As far as we know, there are no constraints on that. You could come up with any sequence and make it. But the peptide bond means that the secondary structures do have constraints. And in secondary structure, what we see are local patches of repeating order, where a pattern of order repeats over 5, 10, 15 residues. And there are two major forms of that we'll take a peek at, the alpha helix and the beta sheet. When you've done that and you then let the rest of the molecule do the rest of its things, overall, that protein will take up a particular configuration in space, and that then is the tertiary structure, the overall packing of the entire polypeptide. If a protein has more than one polypeptide, it can have quaternary structure, which is the packing of the polypeptides in the overall complex. Myoglobin has tertiary structure. Hemoglobin has tertiary and quaternary structure. So the two major examples of secondary structure in proteins are the alpha helix and the beta sheet. Alpha helix, another right-handed helix. They seem to be everywhere in life. And it's the result of multiple hydrogen bonds forming. You can see many, many hydrogen bonds between the oxygen, the carbonyl oxygen, and the amide hydrogen. In the alpha helix, those hydrogen bonds go up and down the helix. In the beta-pleated sheet, they go side to side, spanning the sheet. In both cases, the side chains are going to make a difference to how things overall pack. Obviously, if you put too many big things side by side, that'll tend to constrain it. Beta sheets can be parallel like this or anti-parallel depending on the rest of the configuration of the protein. Some of the common features of the tertiary structure is that if you're talking about soluble proteins, or the bulk of the proteins in the cytoplasm, so most proteins, there will be a hydrophobic core and a hydrophilic surface. That the side chains that like to interact with water will be there at the surface, and the hydrophobic ones on the interior. Some proteins also have prosthetic groups, prostheses, helping things. In the case of myoglobin and hemoglobin, it's a heme group. You can see the heme group more clearly here. This is just the ribbon diagram showing you the 8 alpha helices of myoglobin. Are we at 4 o'clock? 4:20? Okay, we can call that quits.